Okay.
I can let you know when I'm in, Gabriel.
Okay.
I'm in.
Oh, we're getting some echo.
Sorry.
All right. Good morning or good afternoon, depending on where you are. My name is Gabriel Lopez. I'm professor of chemical and biological engineering at the University of New Mexico. And I'd like to welcome you to our SynCell 2021 speaker series. Today we have two talks from Germany. The first one will be by EBERHARD BODENSCHATZ, and the second will be by LADO OTRIN. I will introduce Dr. Bodenschatz here momentarily. I just want to remind everyone that we encourage you to submit questions through the Q&A feature on the webinar. And at the end, we'll get through as many of those as we can. Basically, we have a talk for about 30 minutes by Dr. Bodenschatz, and then we'll have about 10 minutes for questions. And I'll try to keep him to that time. And then we'll switch to Dr. Otrin. Okay, so we are very pleased to have Dr. EBERHARD BODENSCHATZ speaking to us today. To my knowledge, he started his independent academic career at Cornell University, where he rose through the ranks to become a professor of physics. After that, in 2003, he became director and scientific member of the Max Planck Institute for Dynamics and Self-Organization, and he continues to have close ties to Cornell University, where he is adjunct professor of physics and mechanical and aerospace engineering. In 2007, he also was appointed full professor of physics in the George August University in Göttingen, and he is also co-director for the Max Planck University of Twente, Center for Complex Fluid Dynamics in the Netherlands. And finally, I would like to mention that he is a founding and steering committee member of the Max Planck School Matter to Life, which is focused on engineering of synthetic cells. So with that, I will let Dr. EBERHARD BODENSCHATZ take it over to talk about synthonems, building blocks for artificial cilia and flagella.
Well, thank you, Gabriel, for having me. So on my talk, I will summarize our recent work on synthonems. This work is done by Isabella Guido and Azam Gholami, who are both group leaders in my department, and I'm mostly reporting their work. But I hope I can answer most of your questions also. And so I got fascinated of cilia a while back when we investigated together with Gregor Eichele here from the NPI for biophysical chemistry, cilia in the brain. And here what you see in this video is a cut through a tissue in the third ventricle of the brain. And you see these cilia beating along. And this is what's happening right now in our brain, hopefully, otherwise you would be sick. And so the cilia, they're typically 10 microns long. They're cells. These cells are multiciliated, so they have many of these little hairs that are beating. And of course, the flow that is generated by these hairs is mostly in a very viscous regime. The length scales are tiny, the flow velocity is so small, and so the Reynolds number is also very slow. So this is in the highly viscous regime. And so I have to, there we go. And so as you see here is a summary of a few examples of cilia. One is, of course, sperm, which is propelled by a flagellum. Chlamydomosa has these two flagella, which are beating like this, and the cell is basically swimming along. I will use this one in further down in my talk for quite a few things. There's other multicellular organisms that use this, or we have these trachelial cilia in our lung, which hopefully propel all the mucus containing all these viruses out of our lung and gives them away so we don't get sick. And then here you see a cilium beating in the brain on the way on the right side down there, where I just show you the video. This is by Christian Wissendorf here in my institute. And so what cilia are everywhere in the body. Without cilia, we would not have, it would not be possible to have a baby, not only because of the sperm, but because of the ovum, the egg in the uterus is transported by cilia also. And so there's many, they have some in the kidney. They're everywhere, and the cilia they can be sensing cilia, or they can be motile cilia, and I will only talk about motile cilia. And so in the brain, for example, here you see a little cut through the third ventricle of the brain. And what's most exciting about this is you have these ependymal cells. These ependymal cells have these little cilia. And then down here you see a typical transport on a cutout of this tissue. And so this is about probably 400 microns across, 500 microns across of a millimeter. And what you can see is you can have very distinct transport in a 2T sheet. So what you do is you put a dye down, and then you can see that the cilia feed in a certain direction, and they transport the fluid in a very directed manner. And this you can also read up in this science article that we published quite a while back. And so on the right side, you see they beat very, very fast. The time scale here is milliseconds. So you have a very, very, very fast beating structure. How are these made? What are these cilia actually made of? So they are ubiquitous in nature. So pretty much every eukaryotic cell has it and can make it if it's programmed to do so. And so what you have is you have a basal body. So you have to anchor one of these little hairs, which are called axonemes. And you anchor them in the basal body inside of the cell. That gives them a direction and also a B direction. And then on the top, you have this axoneme, and inside you have these microtubules. So you have 2 central microtubules and 1, 2, 3, 4, 5, 6, 7, 8, 9 outer microtubule pairs. And these microtubule pairs are connected with dynein motor arms. So these are dynein motors, and these dynein motors act on these microtubule pairs and bend them. And through that bending, the axoneme moves, and when the axoneme moves, it can basically whip like a whip, and this therefore transport this very viscous fluid in a way that it basically goes through the 3D space, then goes back onto the surface, pulls back, and then goes out again. And by this, these microtubules the cilia can actually transport fluid. So how is this done? Well, as I said, here are these microtubule pairs over here. Then here you have these dynein motors, the outer doublet between the outer doublets. And then what happens is you give them ATP. And when they get ATP, they somehow get this elastic structure, which is about 10 microns long, to bend. And through this bending motion, these things start to beat and can transport fluid. Now, as I said before, is you have not just 2 of these little arms, you have, I don't know why I can't go back. Okay, I don't know here. What you do is you have these 2 here, there's another pair, and then in between you have these dynein motors, but it continues like this. And actually how these beat is understood to some extent, but not really fully. And what you definitely need is these 2 central microtubules. If you don't have those, then these cells will not be motile. They will just be sensing cilia. And so these are little, basically, nanomachines, and it would be wonderful to reconstitute these things from the bottom up, which we typically do in bottom-up synthetic biology. And so first I would like to talk about the work by Isabella Guido. She builds synthetic axonemes. So axonemes, as I've shown you, were these structures which are inside of one of these flagella. You have these doublets, and then they're beating. And so Isabella set out to build some of these artificially. And she used stuff originally from the literature. And so what you see here is you take these few building blocks, and the idea is you take microtubules, you take kinesin motors, and very much like a sliding assay, which was already known before. And then you ask yourself, can I build a beating structure with a microtubule and a surface? And so what she did is she basically reconstituted the system where microtubules would grow. These microtubules would actually adhere to one end, so it's not quite a sliding assay. there's little tricks that you can do to the surface so this actually happens, so that these microtubules don't slide along the surface. And then you have here these kinesin motors over here, and then these kinesin motors actually transport and bend this elastic biopolymer, this microtubule, and by this bending motion, you get a certain force, and when the force is exceeding a critical force, the motors let go, and the whole thing will relax. So let's see how that looks like. So here in the gray that you see in the background here is the actual experimental observation, and the green and the blue is a fit to the structure. The red are the force arms that one needs to actually reconstitute this motion that you see over here. So let me play it again. So what you can see is this thing is actually being moving around, and then whoosh, you see all the forces are gone. Then it goes straight, then it goes straight again. The forces bend the whole thing until the force exceeds the threshold, and bang, it lets go again. And so this one can actually analyze. So Andrei Vilvan and Rami Golestanian came up with a model which uses elastic beam equations. And then you have the flexural rigidity of the microtubules, and you put in motors. And then you follow basically, this is the gray, here's the experimental observed structure by a fluorescent microtubule. And the green is basically the best fit one can get by using different forces. And then what you can do is by this, you can see what forces do you actually get. You can also ask, are these forces always tangential or are they perpendicular? So are they along the microtubule or at the right angle? And you would see both of them are quite important. So you have a force which is centered around 0, but minus to plus 10 piconewton. And then you have also minus 10 and 10 piconewtons in the transverse direction. So the longitudinal one you would not have thought would exist, but of course it does, because it basically pushes the microtubule back, and then it starts to buckle, and then also leads to this motion. So this you can read up in this paper by Isabella in Nano Letters in 2019. And if you just click on this QR code, you will see the paper. And so this allowed me to build with this Isabella built a very, very simple synthonim, if you want. It's just one little microtubule attached to the surface at one side, and then motors push it around. But this is not a flagellum. A flagellum, or a very simple axonym, you would like to have go into the third direction. You would like to have it beat out of plane, anchored to the plane, and beating upward, just like an axonym that I show you here on the left. So this is what you want to do. And so what Isabella realized is the most simple structure is, why don't we use just two of these dynein arms over here and attach them with dynein motors? If you have these dynein motors, not kinesin motors, but actually the real motors that are being used to make axonemal dynein, to make an axonym, can we rebuild a beating structure? And so what you see here, so here are these microtubule pairs, or the microtubules, and then you give them ATP, and then they should also beat without having this complicated structure of the axonym. So how does she does it? Well, what she does is she takes a chlamydomonas algae, like the one you saw swimming at the beginning. You can cut this off with some chemical procedure, and then you can denature this whole beast, and at the end you can have little parts of this axonemal structure over here, the little tiny parts that look like this here on the left side in figure 1. And so then what you do is you then grow, like shown over here, you grow your microtubules out of that structure. So down here is the axonemal seeds, and then out of the axonemal seeds you grow microtubules. Of course, they will grow in any random matter, but once in a while you're lucky and they grow next to each other. And then what you do is you take this outer dynein motors, and you attach them, in a manner that is exactly the same as it's done in the real axonym. They're attached in a periodic manner with a distance of 24 nanometers periodicity, just like in the natural system. So what you need is a ruler protein that allows you that these guys actually attach in a certain periodic manner, just as they do in nature. And here you see different analysis done. You can also see this in the paper. You can make an X-ray diffraction, and you can electron diffraction, and you can actually see where these motors are located and what's the periodicity is. So what you now have is you have a two-armed synthonym. So again, synthonym is just a synthetic axonym, and the most simple version of it to be true. And what you then get is you have these outer dynein arms, you attach them in a periodic manner. The distance here is about 30 nanometers. And then, of course, what you can also have is they could be further apart like this, and then the motors are even further stretched, and the distance would be 200 nanometers. So it would be a much larger distance. Now, it's a little hard to really see what you have under normal microscope, but what you get is the following. So with the motors from Japan, what Isabella was able to do was, with her collaborators, she was able to take these. So this is a microtubule over here, and this is a second microtubule, which now has these axonymal dynein motors. And what you can see is that you get this constant flexing going on and the bending and slashing in the system. So how can we understand this? Well, let's think about a buckling instability. So let's take a bar, and you take this bar and you push the bar together. Then what you get is you get just a buckling. If you then stretch it a little less, then of course it's like this. Or if you have two of these next to each other and you slide them, let's imagine you have two elastic rods. You clamp one side with your hand and you slide the other one up. What you will see is that the lower one suddenly will let go and will actually buckle out with respect to the one that supports it. So before we had a surface here, remember we had the motors on the surface. Now we actually have two microtubules. These two microtubules are sitting next to each other, and in between are these motors, and they're being pushed like this. And then one side buckles out, and then finally it lets go and it relaxes. So here you see this. So first you have this situation. You have this very strong force between these two microtubules. Then at a certain force, some of the motors will let go. And then you get this buckling, but there's still some motors left over here. And then you get this motion. And when you do an analysis, you get that this force is 90 piconewtons, but this is also in the paper and you can read it up. So how do we understand this motion that is seen over here? So here you have these two microtubules. In between, you have these axonymic dynein motors. These axonymic dynein motors push the upper microtubule, which is clamped over here, towards the other one. It pushes further and further until the critical force is reached and the whole thing lets go. And with this, you can actually explain this motion very, very well. So with this, we think this is the first time that there's an active beating of a reconstituted synthetic minimal axonym, which I like to call a synthonym. And so you can find this paper here in the literature, just on arXiv. We are still publishing this work. But I find this work to be absolutely essential, because in contrary to what other people have done, is this the first time that you actually have a very, very simple system of two microtubules which extend into the third dimension, which are actually moving with respect to each other and are beating. And this is only possible because we use axonymal dynein motors. With no other motors, we were able to get anything close to that. So it's quite different to where we use the kinesin motors, the double-headed kinesin motors. This is very, very different, which is very close to a sliding assay. This is not a sliding assay. This is very different. And so so in summary, what I've shown you so far, I've shown you this very, very trivial synthonym, which is really just one microtubule on the surface being pushed around by motors. And here I've shown you the promising result of the first synthonym. So we can assemble more complicated structure out of these things over here. Perhaps we can actually reconstitute a real axonym and actually make and understand better how axonyms are actually moving, but more work needs to be done to explain this. So let me go to the second part of the talk, which is about fuel for synthonyms, which is also related to the second talk today that we will hear from Magdeburg. We work together with the Magdeburg group. And so what Asam does is she took axonyms. So she doesn't take synthonyms like the ones we reconstituted. Here we take actually an axonym, which is a demembraned, broken off flagellum of Chlamydimona. And when you activate it with ATP, then they're repeating, as you can see here. And so when you look at that, so what you do is you take this green algae, you cut off one of these little arms, you demembrane them, you put in ATP, you put a reconstitution system for ATP, and then they just keep beating. And you can see the imaging rate here is 1,000 frames per second. And over here, you see the time, they're actually beating much, much faster than the reconstituted ones that we were building before the synthonims. And so if you look at the time evolution of one of these things speeding along, what you can see here we have, it's very beautiful. I mean, I like this a lot, fits a lot to Santa Fe, by the way, in terms of the colors, I think. And so what you see here is you see these, the colors basically give the position of this axoneme in space as a function of time. So red is late times and blue is early times. And what you can see here is that the beating frequency depends strongly on the ATP concentration. So if the ATP concentration falls below a certain limit, you get no beating. And then if you increase the ATP, then you can see that the beat frequency goes up and they're beating faster and faster and faster. So all the way up to about 60 hertz, which is very, so one beat, 60 beats per second. So this is quite large. And so if you want to now do this, it's really tricky, because if I want to have, for example, my synthonims or these axonemes to go forever, I need energy. So I need an energy module. I need to get some energy. And the one way we did this together with a group from Magdeburg work with Tanja Widowic-Koch and Kai Sunmachte and Kristin Kleinberg, what we used is we used biorobosin, where we basically use light and have this as a proton pump, which then makes out of ATP. So we have this bacteriorhodopsin and we have this synthase, and then they put this into a vesicle, and then they also studied how it depends, this energy production depends on the vesicle that they're being using and so on and so on. But what you can see here already is that the ATP concentration, if you have these vesicles, these lipid vesicles inside of a solution, you can see that the ATP concentration just increases. So just to show you that this works, you can turn on the light on and you can turn the light off, and what you see is that the ATP goes up, and then the ATP gets basically it decays because it's being removed, and then it goes in again, and so on. And so what you can do is you can say, well, what do we do? What happens if I put a lot of these vesicles in a solution, in a buffer solution where I have axonemes? Is it possible to actually drive these axonemes forever just by putting light into the system? The answer is yes. You can pretty much run these axonemes as long as you like, as long as you light the system and put light on it. So the idea is, again, that the ATP goes to your dyneins, the dyneins use this for their motion, and then the whole thing beats. And so here's a movie with this reconstituted system running. Azam and co-workers have investigated this very carefully, have looked at the time evolution, have looked at how long could they run this, and so on. And there's no apparent slowdown to be found. They also investigated different lipid vesicles and also the different structures, and have analyzed very carefully what the efficiency is and how it can be optimized. So then one question is, can we make something simpler? And so here Isabella and Azam come into play. And so the idea was, can we use this very simple system of two microtubules that we over here with the kinesin motors, put this whole thing into a small vesicle, which is made in basically a microdroplet, which is in the oil phase. Then can we introduce this energy module over here, and can we then observe what should happen? So what we know is, if we do this in the bulk, we know that as soon as you give ATP to these motors actually will drive the microtubules in such a way that align themselves in a ordered structure into filaments. These filaments then very often make some kind of liquid crystalline-like structure, and the whole thing collapses like a gel. It's an active gel that is collapsing. Isabella Guido has a whole paper on this also. But let's see what happens in these little water and oil droplets. And so what you see is, you can see that this network that is initially there on the illumination is contracting. So now we have to illuminate. We don't give actually external ATP, and then what it sees, you get also inside a flow, and you can see the structure over here. So this is after 0 seconds. This is after about 300 seconds. And what you see, the surface area has gotten much smaller. And what you see, there's this strong contraction depending on the size of the water and oil droplets that they have been using. And so here you see a video of the, well, does it? Yes, it's running, of the light-powered contraction of the microtubule network over here. So this is really light-powered. If you turn the light off, this whole thing just stops. And all this has been published, or is just appeared. It's not really appeared, but it has appeared in this ACS Synthetic Biology article. As you can see, there's the group of Kai Sumpmacher, Tanja Vidavy Koch, and many others over here which work together. Wahid is working here in our work with us. Albert is also Max Planck. Rahil is the graduate student. All this was funded from a EU grant, and they also got the front page of ACS Synthetic Biology. I think it hasn't appeared quite yet. You can see here it says XXX, but the paper is already online and you can read it up at this point. So I'm a few minutes early, but let me close with this. So here you see a picture of my group. So here's me over there. And so here are the most VIPs, the very important persons. So this is Azam and Isabella Guido, who were running this investigation. And you can see I do many other things other than just synthetic biology. I'll be working on COVID-19. We're working on turbulence and transport of turbulent flows. We work on cloud microphysics, trying to understand why it's raining and why it's sometimes raining much earlier than anybody thinks it should. And so we work also on the Earth's climate, and we also work on cardiac dynamics and try to understand fibrillation in the heart. But this was not the topic of my talk here. This was all about synthetic biology. And with this, I would like to close. Thank you very much.
Thank you, Eberhard. That's a fantastic talk and some beautiful results. I want to again encourage the audience to submit questions through the Q&A. We have plenty of time for questions. The first question we have is coming from Jacqueline Delora, and she is asking, how is the beating of the synthonims or isolated flagella dependent on the fluid dynamics of the submersion media? Is it important to reconstitute a physiological system, or in the implementation of a synthetic system, is it possible to diverge from what nature has already evolved?
Okay, so to be honest, what we do is all reconstituted, but I assume it's very close to biology, what's naturally there. Viscosity, probably you can tune as long as the osmotic pressures don't get totally berserk. But you should really ask Isabella and Azam that question. So I'm sure they're very happy to answer your questions. Just send them an email and they will come back to you.
Okay, thank you. Another question she had is, in the light-powered experiments, why are the droplets deforming?
Okay, so let's see. Do we know why? Let me see. So, well, hard to say, right? So what you see there is what you think what might be deforming. Let me go back one slide. So what you might think was deforming is you mean over here, this looks like, yes, it seems to be deforming. Well, very good question. Why would it be deforming? Because we have surface tension. I mean, this would mean this thing has to be anchored somehow to the interface, right? Otherwise, it cannot be deforming. Otherwise, we would assume that this thing is in the middle and would be surface. But they were deforming, so very good observation. I hadn't realized that here they are very round initially, as you can see. And this one is still round, but this guy here definitely looks like it's an egg. Fits very well to Easter. But it seems to be an egg shape. And so, very interesting question. But the other ones you see, so this is a very dense network. They're really right in the middle over here on the bottom. This one is deformed. But one would have to do experiments where these things are really not in contact with each other, with the other oil droplets, I would say. Yes, next question, please.
Next question comes from Susie Atlas, who asks, in the synthonim work, are you planning to try to push further into a more fully biomimetic / artificial regime? And if so, which parameters do you imagine tuning or changing first?
Okay, so clearly I want to do that. So when I got motivated by this idea of making a surface that is transporting fluids in a directional manner, even in some kind of lab on a chip kind of environment. So you could, at least the cilia in the brain, the ciliated ependymal cells that we have in the brain, they're very directional. They are evolving over time. There's directed transport. And this, of course, would be great, right? I mean, for example, have a spray would be fantastic. Somebody has a cystic fibrosis and you just have a little spray. A doctor came to me and said, don't you have a spray that I can spray to a patient and then he's healed? Well, this is, of course, would be a dream come true, right? You have a little spray and you spray in your artificial cilia and suddenly the person has his own cilia and it's pumping again out the mucus and the person's safe. I think we're infinitely far away from that. But what is the next thing we would like? The next thing I would like to do is I would like to anchor these synthonims. And first of all, get a syntonym that beats perhaps a little better than the one we have currently. But we already have axonyms, so you could also say but they're hard to handle. What would be great is to make an array of these and to anchor them and to make actually basal foot. I think the next thing that's needed is to anchor these to a surface and to make sure that they're beating in a certain direction. Because otherwise, how can you get so now we have to work on how to make a basal foot. This is my personal opinion is it might be able to do this with chlamydomonas or something and basically catch the whole animal and use them, but that wouldn't be a bottom-up approach. So I'm happy to hear any suggestions that you might have, but we're trying to capture these with antibodies or by mechanical means. It's not easy, and we haven't really made much success in that direction, but it would be great to make a ciliated carpet. So this is what I personally would like to do. I don't know what Isabella and Azam are after, but this is what I like a lot, is this ciliated carpets. Another one would be to make a vesicle and to attach some of these as a motor, so like a sperm, so have something that actually swimming along. And then to add to it some kind of chemotactic module that actually tells this object to beat in a certain way so that it's moving from A to B in a directional manner. That would be another thing that would be a high hanging fruit, in my opinion.
Thank you. We have a question from Felipe Quiroz, who's asking, what is known about the length of the flagella and its distribution in biological systems? Is the length a major and reasonable parameter to control in the context of artificial cells?
Well, I mean, the length that I'm aware of, they're typically, most of them are of the order of 10 microns. So the ones in my picture that I had, so the one that come from ependymal cells, flagella in sperm is longer, much longer. Then you get also this kind of wave-like motion. You not just have one, like in the chlamydomonas, you just have this one little beat. You never really have a full, you never have a long node, and then the whole thing is like a snake. What controls well, I don't know. What was the question again?
Is length a major and reasonable parameter to control in the context of artificial cells?
Well, I mean, length, I mean, it seems like 10 microns is something that comes about by itself. So you don't have to do too much to get something between 5 and 10 microns. It seems to be sufficient to get some beating. You could possibly make them longer. Is it a big factor? Well, it depends when you do the resistive force theory for fluid dynamics. So when you do the interaction of this object with the fluid, and length surely is a parameter that you have to consider. It will also determine the frequency and so on. So the advantage of these synthetic systems is that you can really, at least we hope that we can basically take the system and make it as artificial as possible, so that we still have the major features, and then try to understand that system, and then go back up and say, what could the final system actually be? So the idea is a little bit what physicists love to do. They say, what is an elephant? What is a cow, right? And at the end is a sphere. So you want to go to the sphere and then work your way up to the more complex animal. But I think with the synthetic biology approach, we could do this. I should also mention, of course, that this whole field here of microtubules and motors goes into the field of active matter. So it very strongly also couples into the field of active matter. And so you get, active pneumatics, you get all kinds of different elastic properties. These are very different materials because, just from the perspective of material science, what I show in the figure here, this gel that you see there, which is sitting in here, that's an active gel. This is not a passive gel, this is an active gel. And so these materials will be very, very different from anything we have been looking at in the past. Getting closer to biology, but perhaps also closer to really synthetic systems that have overlap with biology, but are not really biologically relevant, but might have other technical use.
Okay, thank you. We have another question from Lynn Rothschild, who writes, have you drawn any inspiration from natural biosynthetic pathways for cilia? For example, in the alga Chlamydomonas. Thanks, fascinating work, she says.
Chlamydomonas reinhardti. Yeah, so what we do is we harvest pretty much everything from chlamyd. So we are growing chlamyd down in the lab. We have a little aquarium, and then basically things get harvested that way. Because you need the motors, you need the microtubules. We grow differently, but you might need the seed crystal, right? This is what I was showing here, where Isabella is growing the things out of basically a seed crystal, if you want. So when you go here, back here, right? So this artificial basal body, if you want, where you grow the microtubules out of this. So this is also from chlamyd. So this is stolen from chlamyd. So we are still using biology quite a bit. So this is not really fully bottom up. There is a little bit of use of biology, not just chemistry and so on. But in this case, this was the best. It's amazing that Isabella could do it that way. I'm still amazed that this works.
A related question is from an anonymous attendee who says, who asks, is the faster beating of the flagella from this Chlamydomonas compared to the synthonims a function of the number of molecular motors and number of microtubules involved?
I would say this is a very interesting question. Very interesting question. Well, clearly, I would suspect yes, but this is now a pure guess. It's much faster, right? It's much, much faster. 60 hertz is quite a speed, to be honest. And we have, of course, multiple of these structures that could give you an increase in frequency, right? If you interleave these in such a way that some basically are recovering while the other one is contracting, they could give you an increase in frequency. I don't know. Something I have to think about, but a very interesting question for sure. Thank you.
Sure. Well, thanks to the anonymous attendee. Okay, we have one more here. And I think maybe you've addressed this, but I'll go ahead and ask it. It's from Ana Relimpio. She asks, did you try reconstructing the microtubules in the axonemes of the algae, only cutting its end?
Oh, she wants to, I see, she would like to grow out at the end. Talk to Azam and Isabella.
Okay.
I think the control is not quite that good, but I might be wrong. So please talk to Azam and Isabella.
Okay. Well, I would encourage anyone in the audience who has further questions to definitely email the people that Dr. Bodenschatz has mentioned, and also Dr. Bodenschatz, if you have any questions.
Just send me a copy so I see what you asked.
Okay, Eberhard. Well, thank you very much for the very interesting talk. I will introduce the next speaker in just a few moments here. Okay, so it is my pleasure now to introduce Dr. LADO OTRIN. LADO, do you want to share your screen? Great. Fantastic. He is coming from the Max Planck Institute for Dynamics and Complex Technical Systems, and he is going to tell us about his research on artificial mitochondria from the bottom up. Please, LADO.
Okay, well, thank you very much and happy Easter to everyone. So I conduct the research in a group, Electrochemical Energy Conversion, headed by Tanya Vidakovic-Koch, which is part of Process System Engineering, headed by Professor Kai Zumacher. And our institute is located in Magdeburg, East Germany. So inside of a Maxi Bio consortium, our task is to provide energy regeneration to the artificial cell. And as you heard from the example for Professor Bodenschatz and some other speakers, pretty much all of the synthetic or biomimetic modules require ATP to function. The flagellar movement, ciliar movement, actin polymerization, various metabolic conversions, everything requires ATP. So if we mimic these functions, this ATP price is also mimicked along with it. And in nature, there are many ways to produce ATP, but the yield is much different. And for example, one can see that a cellular respiration, being the most efficient ATP scheme, is much more efficient than some other ones. And it is no surprise, actually, that most of the modern energy regeneration systems published in recent years can be seen featuring enzymes of phosphorylation. So this is either photophosphorylation driven by light or oxidative phosphorylation driven by some reducing equivalent, for example, NADH. How does this work? So what is required to mimic these functions? It's actually pretty simple. You can reduce the number of components to the essential ones. So in this case, we need membrane from which we can form vesicles, which enable the formation of gradient, which can then be used to drive ATP synthesis by the ATP synthase. How this gradient is formed is a bit more versatile. So basically you need a proton pump. And in the case of oxidative phosphorylation, it's a chemically driven proton pump. In the case of light-driven systems, it's light-driven proton pump. And then, of course, you need electron shuttle to transport electrons between these enzymes. And last, you need one of the enzymes to connect to the metabolism or to connect to some other biomimetic module. Usually, this is NADH dehydrogenase because NADH is the most common reducing equivalent. So what exactly are we building? If we look at the enzymatic composition, we're building artificial respiratory chains. This is our energy module. So the most basic version, so the essential components here are the ATP synthase, vesicle, and the proton pump. And then in the next step, we are adding NADH dehydrogenase, which, as I mentioned, enables us to connect this module to metabolism. Now we have two options here. One is the integral membrane protein, and the second one is water soluble. And the difference here is that the integral one is another proton pump. So in theory, this should also contribute to a proton gradient, and the module should be more active than the water soluble variant. Now, when we connect artificial respiratory chain with the metabolic cascade, we call this minimal artificial mitochondria. So the logic is pretty much the same. Instead of the Krebs cycle, we have metabolic cascade. Instead of the respiratory chain, we have artificial respiratory chain. And then finally, if we want to have the entire finished organelle, we also encapsulate this reaction into the outer compartment, which also contains transporters for the substrates and for the products. Now, there are several challenges connected to the construction of artificial mitochondria, and I will talk about a few of them. So one is that, for example, this energy module must be stable for quite a while. Ideally, this should function for a couple of days, couple of weeks, perhaps, in the context of artificial cell. So one of the things that we must do is increase the functional durability of this module. Another thing is that all these respiratory enzymes are known to produce reactive oxygen species. So that means that if we want to have this stable system, we need to stabilize it further because reactive oxygen species would react with, let's say, lipid membrane, which causes the lipidation of enzymes. So basically that means that the enzymes would fall out of the membrane, which deactivates the enzymes and destroys the module. Now the second challenge is that all these enzymes need to be inserted with correct orientation. So orientation definitely matters here. We need them to pump protons towards inside of the vesicle, and then this gradient can be used by ATP synthase to produce ATP on the outside of the vesicles. And also, of course, all of these copies of the enzyme need to be in the same vesicle at the same time to have the fully functioning artificial respiratory chain. Then the third aspect to this is that we must be able to couple our energy module with other biomimetic modules. So, of course, we need to match things like osmolarity, ionic strengths, pH, optimums, and so on. And lastly, we need to provide continuous oxygen supply. So the oxidase here is using oxygen as a final electron acceptor. And in the absence of breathing, lungs, and bloodstream, this oxygen is not easily replaced. So if you imagine these reactions happening in about, let's say, 0.5 milliliter volume, oxygen is depleted extremely fast, and then the activity of the module is limited by diffusion. So now the first aspect is how we increase the functional durability and stability. We do this by basically supplementing lipid vesicles with polymer. In our case, this is graph copolymer PDMS-GPO to form hybrid vesicles. So these vesicles, there are a couple of advantages. One is that the lipids, some of the proteins require specific lipids to function, and then this architecture can accommodate for these requirements. And the second thing is that we notice that once the proteins are inserted in hybrid vesicles, the permeability of the membrane actually decreases, which is extremely beneficial for our case. And then the second option is that we replace lipid membranes entirely by polymer membranes. As you can see from the cryo-EM images, the membrane thickness is basically very similar. The fluidity is very similar. So we postulated that the membrane proteins will behave in a very similar fashion. They will be highly active in these artificial membranes, which so far wasn't the case, for example, in block copolymer membranes, which are much more stiff. For example, then we checked if actually this polymer is protecting the respiratory enzymes against reactive oxygen species. Here on another cryo-EM image, you can see a lipid membrane which reacted with ROS. And these indents are actually damage done to the lipid bilayer. And then in the next step, the enzymes will be removed from the membrane and module will be inactive. So what we notice is that if we treated lipid vesicles with, for example, ascorbate or ascorbyl-free radical, almost all activity is lost of respiratory enzymes. Now, if you insert these enzymes in hybrid vesicles, so a blend of polymer and lipid, ascorbate basically has no effect on the system, while the ascorbyl radical still destroys the activity of the system. But if you insert this in pure polymer vesicles, almost all of the activity is preserved. And now finally, we measure the activity of the energy module after 11 days, and we see that the activity was prolonged in hybrid and polymer vesicles, while the lipid vesicles were completely inactive. So now that we have this module chemically stable and functionally durable, we inspected what is actually happening with the membrane proteins. So how active is our system? So how we do that is we use the traditional detergent-mediated reconstitution. We prepare solubilization profiles with different detergents, reconstitute membrane proteins with different setups, measure their activity. And basically what we can see is that, for example, the activity of BO3 oxidase, the proton pump, was preserved across all the systems. But if we reconstitute BO3 oxidase and ATP synthase, the ATP synthesis in polymerosomes is only about 1/3 of that in liposomes. So some activity is lost. And the question here was why. So we analyzed the reconstitution efficiency, so the insertion efficiency of respiratory enzymes and their orientation when reconstituted with different setups, so different detergents at different detergent concentrations in different vesicles. So without going into too many details here, what basically we can conclude from this is a comparison of the reconstitution parameters for BO3 oxidase and the ATP synthase. And what we can see is that the optimal reconstitution for one of the enzymes in one of the systems is definitely much different than for the other enzyme. and also between the systems. So there isn't a single optimal reconstitution protocol, and some of the function must always be sacrificed if one is intending to reconstitute more than one transmembrane protein. If we think about this, that is actually expected, because only one setup, really, one configuration of these enzymes will lead to very active system. Some will lead to some activity. So basically, if we have two proton pumps and one is pumping in one direction and another in the opposite direction, the net proton flux may still be enough to drive the ATP synthesis, but the ATP synthesis will be impeded. But the most outcomes are negative, so there is no ATP synthesis. So we're trying to find a way around it. And since we know that we can optimally reconstitute a single membrane protein, we were thinking, okay, well, we can optimally reconstitute single proteins, co-reconstitute snare, fusogenic proteins, and then fuse this system to form basically a system with ideal configuration. So what we did here is exactly that. So we reconstituted ATP synthase optimally with one half of the snare pair. We reconstituted biotrioxidase with another. We fused the system and measured the ATP production. And remarkably, basically, what we saw is that if you compare then the final activity of the system in lipid, hybrid, and polymer vesicles, not only that now we can match the activity in hybrids and polymerosomes to the activity of liposomes, but we can even surpass it in polymerosomes because polymerosomes fuse more efficiently. So the second thing is that how can we facilitate coupling with other modules? And one of the things that we must be careful about is osmotic stress. So again, we are trying to measure here how our, what the activity of our energy module is under different osmotic stress conditions. So here we see the size of the artificial respiratory chain vesicles measured by DLS. And when we would induce hypertonic stress, the vesicles would shrink, but the activity is nevertheless preserved. Now, the other part is more problematic. So what happens when the vesicles swell? So again, we see drastic swelling when we induce hypotonic stress of 600 milliosmol difference. But even though the vesicles are drastically expanded, they're still active, so they do not burst. So towards this end, we stabilize them in high sucrose solution and added some other stabilizers. So this means that we can couple this module with dynamic module, so with another module which has dynamic range of at least 600 milliosmoles per kilogram. So now to the example of the actual coupling. So here we couple our metabolic cascade, which is catch cycle, published by the group of Tobias Erb. This is carbon fixation pathway comprising 17 enzymes from 9 different organisms. And the idea here is that the formate, a simple organic molecule, would be used as a sole source of carbon and energy source. So there is one ATP-dependent reaction. Our artificial respiratory chain here is energized by formate, which is converted to NADH and fed to the respiratory chain, which produces ATP, which is then used in this one reaction. And what we saw is that at the beginning, the activity of the catch was pretty much the same to the controls, but then it decreases a little bit, and the overall efficiency is about 30%. So we wanted to check whether the problem arises from this ATP-dependent step, or it might be just some other problem due to the interactions of vesicles with this enzymatic cascade. And indeed, we saw the accumulation of 4-hydroxybutyrate, which is a substrate of this ATP-dependent reaction. So we saw that the ATP was insufficient in the system. Now we are looking into this, and what we did is we measured the ATP production by the artificial respiratory chain over 2 hours. Like that was the runtime of this coupled reaction. And what we saw is that at the beginning, the ATP synthesis was extremely high, and then this dropped. And this, and then in the next couple of hours, the ATP synthesis was much slower. So we saw that something was happening with the module there. And when we measured the oxygen concentration in the system, basically we saw that the module is so active that the oxidase in the system is depleting oxygen in less than 4 minutes. So what we did is we wanted to design some backup, some auxiliary oxygen regeneration system. which would oxygenate this reaction sufficiently to drive this coupled reaction. And towards this end, we use calcium peroxide particles, which are dissolved in water. They release oxygen, but at the operational pH, they also release hydrogen peroxide, which we then couple to catalase that converts the hydrogen peroxide to oxygen and water. So this reaction shows just the oxygen release by these particles. So this is the oxygen release without catalase, and with catalase, we saw that with catalase, basically, we saturate the water solution. And indeed, when we added these particles to the artificial respiratory chain, we drastically increased this fast ATP synthesis rate for several minutes. So now just quickly, what are some next steps, what we would like to do with this system? One of the things we want to make most of these steps automatic. So we would like to express these membrane proteins cell-free in microfluidic setup, fuse the system or assemble it through fusion, and then also add other biomimetic modules to form fully functional synthetic cells. So the other thing we would like to do is complete the actual mitochondria. So now we have the couplet reaction and the artificial respiratory chain. And what we are working on now is encapsulating this module in the outer compartment and controlling the substrate and the product exchange. And lastly, we would like to attach these vesicles to the scaffold, which contains fibers with embedded calcium peroxide particles. This artificial scaffold would release oxygen and oxygenate our module. And with this scaffold, we can also attach the respiratory tissue to, let's say, reactors and use it in a bioproduction facilities. So with that, I would like to thank our collaborators and you for attention. Thank you.
Thank you very much, Lado. Fascinating work, very, very systematic. Again, I'd like to invite people to submit questions on the Q&A. I am not seeing any questions so far. Here's one from Jacqueline Delora. She says, have you thought about developing a synthetic system that uses, for example, nitrogen rather than oxygen as an input? Would this be experimentally feasible?
Definitely. So my personal ambition here is to go more into, let's say, space science with this and to add some metabolism that is not so biologically relevant maybe, but it's more relevant to extreme environments. So with respect to this, whatever we can fed to the artificial respiratory chain, we can use it to drive the respiratory chain. So I think that this would be possible, yeah.
Great, thank you. Very, very interesting. Another question here. Felipe Quiroz is asking, could you comment a bit on where you are now with regards to ATP/energy output and the needed scale up to match the energy demands of a minimal artificial cell?
Yes, absolutely. So what we are reaching with artificial mitochondrion is up to a few millimolar range, which basically means that we can drive any physiological reactions with this. So the catch cycle alone is, I think it's high micromolar range, about 600, which we can drive. Basically, we can drive most biological reactions. I think all of them that I saw so far, which have clearly stated the ATP requirements.
Just to follow up, have you compared, for example, your production capability for ATP to that of, say, the Craig Venter minimal cell that we heard about last week?
Could you please remind me what was their output?
I don't know what it is. It's just it's their minimal genome cell that they've built.
Yeah, I don't remember. Sorry.
Okay, thank you.
Mm-hmm.
Let's see. Here's another question from Idira Macias. Very interesting talk. If I understood correctly, No microfluidics experiments have been performed. I have read some papers where various modules have been integrated together, including the mitochondria to generate ATP. What makes your system different?
So basically in these one or two systems, this is published by our collaborators, so Professor Erb. They used either isolated mitochondria or isolated chloroplasts. So what we do is we created the system bottom up, and we can tune the composition exactly as we would like it to be. This is the difference. And the difference is also that we can build more stable system. And at some point we are hoping to build more efficient system by reducing the redundancy and by controlling the protein orientation better. So the person who asked the question was correct. We haven't done anything in microfluidics so far. So at least I personally haven't done it, but our collaborators, so the group of Tom Robinson, and also one of my colleagues tried to encapsulate part of the catch reaction, which was done successfully. So we are quite optimistic that we can assemble the mitochondria in microfluidics in the next half a year or so.
Okay, thank you. Mario Mencia asks, he comments, excellent work. He asks, I didn't get why the coupled ATP yield was higher in the liposomes than in the polymerisomes. Did you actually observe different orientations in both systems?
Yes, yes. So we observed not only different orientations, but the main issue here is the reconstitution efficiency, so the insertion efficiency of ATP synthase. So just a second. So for example, if you compare the ATP synthase insertion in liposomes, the most active system is the one constructed with sodium chloride, that is mediating detergent. And if you compare this activity to the most active system in polymerosomes, constructed with OG, we saw it's about 40%. So since only 40% of the ATP synthase in polymerosomes is ending up in membranes compared to liposomes, the overall activity is also much lower. So this is what we're trying to increase now, or that we increased by using fusion to assemble the system instead of co-reconstitutions.
Thank you. Susie Atlas asks, well, she comments as well, very impressive work. She asks, since this is such a complex system with many moving parts, have you encountered any unexpected consequences in this system that required significant efforts for mitigation, or which suggest new avenues for synthetic engineering?
Yes, I think that every step of the way was pretty painful and full of challenges. So one of the things is definitely how to control this orientation. That's why we switched to fusion. One must understand that the fusion of polymer vesicles is basically, it's very limited because they're so rigid and so inert. And there are like 5-6 examples of polymerosome fusion and no example of lipids or protein-mediated polymer fusion. So we did it for the first time. And we had to figure out how to reconstitute membrane proteins along with snare proteins. We also spent a lot of time on creating the oxygen regeneration system. This is also not so straightforward because the particles need to be distributed quite evenly to oxygenate solution evenly. And since they are not coated, they also release oxygen pretty fast. So that's why we would like to embed them into fibers to have the control oxygen release at the control rate, kind of like respiration. And of course, the construction alone. So when we are adding new enzymes to the artificial respiratory chain, the complexity increases exponentially. So it's not the same thing to add third enzyme than it is to add second enzyme, just in a nutshell. But there are, of course, many different challenges.
Okay, let's see. Azam Golami asks, in slide 12, why do you observe drop in ATP concentration after 8 minutes, even with enough oxygen?
Basically, in this coupled system, so in this case, if she's referring to this case, we haven't added catalase because of the background in luminescence measurements. So that's why also the whole signal is more noisy because the peroxide is killing the enzymes. So I think that this drop that we observed is combination of enzymes slowly dying, and also the second part is that concentration of particles that was added was so minuscule to still enable these light-based measurements that the oxygen is released much faster than, let's say, in these experiments when much more particles were added.
Okay. Final question, somewhat related, from Felipe Quiroz. What do you know about how the polymerisomes prevent reactive oxygen species-induced damage to the key ARC components?
So basically, we believe that it's mainly due to prevention of delipidation. So the polymer membranes remain more or less intact. So that means that the integrated enzymes are also protected from the ROS, and also they don't get ejected from the membrane. We tried treating just proteins, so just enzymes with reactive oxygen species, and we saw that they're also deactivated. So it means that the thickness of the membrane alone is enough to protect them partially, but mostly it's due to the lack of delipidation.
Okay, well, thank you very much, Lado, for a great talk, and thank you also, Eberhard, Dr. Bodenschatz, for a wonderful talk. I am going to now introduce the talks that we'll have next week, if I can pull up the right slide. Let's see here. For some reason, yeah, this is the right slide. You should be seeing a slide that indicates that next week we will be hearing from Professor Felipe Garcia Quiroz. He's from Emory University and Georgia Tech, and he'll be talking about membraneless stimuli and stimuli responsive organelles. He's been very active in the question and answers sessions that we've had, so appreciate that, him bringing his insights, and we get to hear about his interesting work next week. We will also hear from Cesar Rodriguez Emenegger from Leibniz Institute for Interactive Materials on superselectivity in synthetic protocells. So thank you all very much for attending, and we look forward to more discussion next Monday, April 12th.